Right now, we'll be talking about bacterial resistance and immunity. Bacterial resistance and immunity. Right now, there are a lot of researches, a lot of shouting up and here in the microbiological world that bacteria are gaining resistance against different diseases. This novel strain of whatever that has been overcome before has come out, and then it has been able. It's not responding to the drugs again. This is getting deadly. Mortality rate is increasing. Morbidity rate is so high, and treatment efficiency rate is so low. Why? Definitely. Do you think the bacteria wants to die? Do you want to die? You are shouting because you don't want to die. So do you think bacteria want to die? They don't want to die as well. So they have bacterial resistance because they have immunity. Now, let's forget about, I would say that it's not actually fully the bacterial fault for entering into our body system, because they didn't come and meet us. We went to meet them where they are. You get? They are not as motile as we are. You get? And their motility is restricted to their habitat. You get what I'm saying now? So it's not fully their fault that they are infecting us. It's our fault for them infecting us. But let's not take it away from there. Now, after they've now infected us or whatever they've entered our body, we now use drugs against them. The drug kill them, and then suddenly they fought back, and then the drugs does not work against them again. Why? That is exactly what they are. That is why they have immunity. We have immunity as a big, complex multicellular organism because we have cells that are doing what? That are performing things like phagocytosis and some other things. And at the same time, each of every cell has their own immunity potential. Aside the normal immune system that actually fights this. There are some infections that is internally into a cell, and the cell repairs itself back, and the phagocytosis does not consume it. That is individual cellular potential for immunity itself. You understand what I'm saying now? So bacteria have their own cellular immunity too. You get this immunity has different ways. Now you hear bacteria can resist extended spectrum beta antibiotics, and they can resist ordinary antibiotics. They can restrict antibiotics of different level using different mechanisms, like the efferent pump mechanism used against things like tetracycline family. You get extended spectrum beta lactamase used against extended spectrum beta lactam ringed antibiotics. There is something like beta lactamases that is used against the normal beta lactam ringed antibiotics, and many more like that. These bacteria don't want to die. Then how do they actually gain this immunity? It's simple. After you've actually exposed them to the drug, the first thing that happens is fitness selection. Now, fittest selection. When fittest selection happens, the strongest among them is selected. If all of them are not strong enough to withstand the drug, they all die. Abi? Yes, you clear them out totally, but not so totally. Some of them still survive. Some survive by hiding, by not getting exposed. When you use drug on antibiotics in the lab, it kills everything. But when you use drugs internally, what happens is this: your cell, the cells that start dying, right? Then some of the cells hide themselves within your own cells for protection. So it means those drugs does not get to them. When the drug does not get to them, but they've been exposed to it, but not to a cedar level, they retain memory of such attack by taking a sample of the drug you gave that you used. They will take a sample of that drug, they will swallow it, they will keep it with them, and they will keep being stressed, but not dead, until their cells have been successfully created a new product that can be used against these antibiotics. And it's simple. What happens is this: once they are exposed, they take in the antibiotics. When they take it in at a non-cellular level, they hide themselves, they escape. You get? When they escape, although it's not totally conscious, but that's how it happens. So when they escape into and hide amongst other cells, it gives them time, or maybe during the process they were excreted before the cells could kill them. You get, like, once you excrete one before the cells could kill them, then during that time they are not dead yet. They may be less motile, so you will not see them doing much, but they are not dead yet. So at that point in time, they will now start using their gene. Their DNA contains instruction on how to produce every protein the organism can produce. But this time around, it's now it will use all the protein against it that it has produced before. If it doesn't work, it will now start recombining itself by doing it will do replication in a recombination way. You get, meaning that replication will be apt, and then it will be recombining and testing, depending on how sophisticated the program in the DNA for genetics recombination is. Some may be very, very accurate and functional, where immunity is very, very fast. In that case, what will happen is that once it comes in, it gets one that is closely related, another one that the next one, you to make sure that from the experience of the first one, you create a more accurate one like that. While some other ones, you just keep randomly creating different DNA strands that will be different proteins rather. You get, using that recombinant DNA fragment to keep creating different protein to fight that organism. So once it has chemical, once it has successfully been able to fight that chemical, what happens next is simple. Now, the last memory of the DNA, recombinant DNA, that produce that chemical, that successfully beat that chemical in chemical reaction, will be saved, and that will be the next vaccine or immunity potential of that organism. That is why that drug will not be able to work against that organism again at the cellular level. Now, that saving that memory of that DNA strand has two methods. Method one, at a point in time, a lot of the when the chemical is inside, there is a calculation. A lot of the protein effective against that chemical will lose concentration very fast, because they are the one actively binding with the waste product and inactivating them. It's the toxin, and it's inactivating them. So when that is happening, the body itself, the DNA, notes the one that is lowest in concentration after producing all of them in a known concentration, which is equal concentration. Then it now do what? It now stores that one that concentrations drain very fast. It stores that into it. Now does it does reverse transcriptase, meaning that protein is now reversed back to DNA strand. And once it's reversed to DNA strand, it's stored as a temporary DNA, which is alternative DNA that you see in most advancing bacteria. That will just say DNA with a. I've forgotten what they call it. I think Andeli or whatever. They call it one name. You get, which is an alternative DNA. You get, so it saves it with that, in that form. You get what I'm saying? Pending the time it has fully confirmed that it will work against that bacteria before it becomes a full part of the DNA of the organism. And during that process, once a bacteria has been able to successfully get that chemical, what happens next is the sexual reproduction I explained, where they now conjugate with each other immediately. Conjugation is always during bad times. You get, during dangerous times, that is when bacteria conjugates. You get what I'm saying? So they now start conjugating immediately. You get, in order to send the new information that kept them alive to the ones that are open to the risk. All of them now send their genetic result of all the methods they've used to combat that bacteria to each other. Then they now use it to form another gene, which is called immunity DNA strand. You get, that strand is now added into the immunity DNA codons of the DNA after it has actually been able to achieve favorable condition. So after it has now achieved favorable condition, it now starts replication of a new strain that is resistant to that bacteria. That is how it works. So on this note, I wait. The first thing is species selection, which I've explained. I use this to explain gene with combination. When I talk about recombinant DNA and all those stuff, then I now finished everything in conjugation, which is as written here. So that is the stage. That is how resistance happens. So when they conjugate, they now spread it amongst themselves that are still alive. You get, they fight and then keep reproducing themselves with, they fight for life, after which they now start reproducing themselves when the environment becomes faithful and okay back. So at that point in time, whatever you now use that drug on, you not even tamper with them again, because immediately it happens, they have a library for it that will counter it immediately, and boom, they're still alive upon how you try to kill them. You get it? So that is how it works. Thank you. God bless you.